Well, welcome. So today, well, first of all, my name is Aaron David. I'm the director of plant ecology. And it is my pleasure to introduce our distinguished seminar speaker, Dr. Anny Chung. Anny is the Haynes Family Associate Professor of Plant Ecology at the University of Georgia, and also holds an appointment in the Department of Plant Pathology. Her research interests focus on plant microbe interactions and their effects on plant competition, coexistence, commonness, and rarity. She received her BA from Washington University in St. Louis, her PhD from the University of New Mexico, and also was a postdoc at Utah State University before starting at UGA. Anny has an impressive funding record with a major grant from NSF and a giant grant from the Department of Energy, and papers in Proc B, Tree, Journal of Ecology, Ecology, and a Tansley Review in New Phytologist. I've known Anny for a long time, and I'm very excited to finally have her out to Archbold. Anny and I have a lot of things in common, including research foci, such as plant population modeling, fungal endophytes, mycorrhizal fungi, and the occasional plant pollinator network project. And then more to the point, we cut our teeth together in ecology in the same research lab at WashU. Although we actually met long before that, we were both part of the same multicultural mariachi band. To this day, Anny remains a virtuoso violinist, and while me on the guitar, not so much. So today she'll be talking about water, fire, air, and earth, plant microbe interactions in the changing world. And leave it to Anny.
Thank you for that introduction. I was like 95% sure that was going to come up, and I was correct. That's what happens when you know someone for almost 20 years. Thank you all for having me out to visit. It has been such a wonderful time and so fun to finally see this ecosystem and visit Archbold, a place I've heard about for so long. And so today I'm going to talk broadly about several projects that we have going on in the lab. So it's, instead of kind of going deep on one thing, I'm going to go broad and shallow sort of. And they all kind of tie together in terms of thinking about how changing climates and other global change factors alter plant microbe interactions at all these different levels of ecological organization. And so we can start, for example, at the individual level. So there are various different things that could happen at the individual level when you think about plant and microbial interactions. For example, you can see changes in antioxidant production from the plant in response to something like drought stress, which then impacts its interactions with fungal endophytes. You can think changes in terms of pathogenicity in the on the microbial side, and changes in its ability to do nutrient acquisition and deliver those nutrients to its plant host. And then these individual level responses kind of all add together and lead to, oh, here's another one, changes at the population level that can result in the reordering of species abundances in a community, right? Some populations could then increase, for example, this willow here, in relative abundance due to a variety of factors, whereas other populations may start decreasing. And then at the community level, then you might start seeing species losses and immigrations. And so, for example, extinctions due to, for example, loss of an essential symbiont, or invasions, for example, the famous pine ectomycorrhizal symbiosis, where you see a lot of pine invasions in the southern hemisphere that's been facilitated by the introduction as well of its mycorrhizal partner. And so work in my lab kind of spans these different levels of organization. And so today I'm going to talk to you about these four little vignettes, which I've kind of organized in a semi-elementally. And this, the first thing we're going to talk about is water. And so I'll show you a little project about the temporal dynamics of drought effects on soil microbes. And so when we think about what happens to plant microbe interactions when water slowly becomes unavailable, there are a few different things that we may want to think about. Well, first, the direct impact on the plant host, right? We all know water is essential for plants. I'll be pretty in agreement about that. And what happens when water becomes less and less available one of those really key things that relates to microbial interactions below ground is the role of root exudation. And so root exudates are these kind of small molecules that plants continuously release out into the soil, sometimes actively, sometimes passively. And they act as both signaling and resource compounds for soil microbes. And one of the things that has been kind of really been a recent focus in the literature is understanding the role of root exudation in mediating the impact of drought on plants, maybe via the recruitment of microbial mutualists that can help plants withstand more drought. However, one thing that we've noticed in the literature is that there are many dimensions of drought. And one thing that's important is the duration and intensity of drought, which are often confounded with the age of the plant. And I'll talk a little bit more about that in just a second. And so my student Xiaomi became really interested in this idea and sought to kind of design an experiment where you can actually tease apart all these different dimensions of drought. And so first, she wanted to understand the different intensities of drought. And so this is fairly easy to understand. You've got extreme drought, you got moderate drought, you got control where it's well watered. And these just relate to different percentages of reduction in water. The next thing was slightly more difficult to think about. And so she wanted to understand the impact of drought duration, so the length of the drought on plant microbe interactions. But if you started the drought at all the same plant age and then stopped the drought at different times, then that treatment is confounded with plant age. And that is often how these experiments can be done in the literature. And we don't have a lot of good understanding about how those two factors can be kind of unconfounded. And so she designed this very complicated setup then, where we had plants at different ages with different lengths of drought that are started at different ages, essentially, right? And so with the oldest plants, they experience both 7, 14, 21 days of drought and control. And then the youngest plants, obviously, we can only expose them to the shortest amount of drought. And so with this semi-factorial combination of treatments, we're hoping to be able to tease those two things apart. And so we conducted this experiment on sorghum. And so sorghum, the reason we focused on sorghum is one, we had a different set of experiments planned associated with a grant with the DOE, who's very interested in sorghum as a potential biofuel crop. And so one of the reasons why sorghum is attractive as a potential biofuel crop is that it is known to associate very strongly with mycorrhizal fungi and also potentially has the potential to be quite drought tolerant. And so another reason to investigate drought in this particular species. And so with this particular experiment at harvest, we measured plant biomass, and we also sequenced the roots of each of these plants for 3 different microbial communities, for bacteria, for general fungi, and also specifically for arbuscular mycorrhizal fungi. And so Xiaomi hypothesized that these different impacts and dimensions of drought should have different effects on these different microbial communities. And so what we found was that plant age actually interacts with drought severity to alter root microbial diversity in interesting ways. And so I'm going to show a series of figures. In these figures, the x-axis is just going to be plant age. And so youngest plants at harvest, oldest plants at harvest. The y-axis is just going to be Shannon diversity. And then the colors relate to drought treatment. So control, extreme, and moderate drought. And so when we looked at bacteria, for example, you can see that drought severity has an impact on diversity. So the more extreme the drought, the less diversity there is in root bacteria. However, there isn't as strong a decrease with age. However, when you looked at the 2 fungal community data sets, the effect of age is reversed. And so with fungi and AMF, what we see is that their diversity actually increases with plant age. That's an important thing to keep in mind before we see the next thing. And in their case, For general fungi, there was actually no effect of the different drought intensity treatments. Whereas for AMF, it reacts to the presence of a drought, but the relative difference between extreme and moderate drought was not significantly different. Okay, and so next what I'm going to do is take away the control treatment and just look at the impact of the length of drought for the extreme and moderate drought treatments. And so now along the x-axis is the length of drought, and these are now averaged over age. And so the really cool thing that we found is that plant age and drought length had actually opposing effects on microbial diversity. And this is the most, it is kind of across the board with these different communities. And so for bacteria, fungi, and AMF, we saw that as the length of drought increased, you got decreasing diversity in each of these communities. And the important thing that I want you to keep in mind is that we saw an increase in diversity with plant age, right? And so what this is telling us is that if we hadn't been able to kind of disentangle the effects of age and drought length in this particular experiment, what we might have seen instead is just a flat line of drought length, right? In terms of its effects on fungal diversity. And I think that was really cool for us to be able to disentangle those opposing effects of temporal dynamics on the assemblages of these different microbial taxa. Next, we're going to look at composition. And I am not expecting you to actually kind of go through every single little panel here and get something out of it. These are ordination plots. And so each point, just to kind of orient you, refers to one individual sample of a microbial community from a single plant. And so the panels are arranged in increasing plant age with different colors for drought length and different shapes for drought severity. It's a lot to keep track of. What I want to just overall convey with these figures is that the composition of all three microbial groups were significantly affected by all the different main effects of drought length, severity, and plant age. But what we found the most interesting really was actually in the spread of these points, or what we like to think about as beta diversity. So turnover diversity, essentially how different within a treatment group are the different samples among that group. And we saw some really interesting patterns here with regard to the length of drought. And so the y-axis here is now the distance to the group centroid for each cloud of points that relates to the combination of those treatments. And what we saw was that longer droughts had homogenizing effects, so decreased beta diversity on bacteria and general fungi. So in the longest length of droughts, especially, you have the most, homogenized group of communities, right? So suggesting to us that these long droughts are a strong selective force on the community assembly of bacteria and general fungi. Whereas AMF kept going up. So the longer the drought, the more distinctly different each community became with the AMF. And so that's really interesting. And Xiaomi has since followed up with some experiments to consider priority effects in AMF community assembly. And we are seeing actually quite strong priority effects that are shown by AMF that we don't find in bacteria and other fungi. And so I think this is kind of a pattern indicative of that, which is these droughts kind of set them on these stochastic trajectories depending on priority effects. And then the longer that drought, the more distinctly different these communities become. And so just wrapping up this first little vignette real quick, what we saw was that these temporal dimension is really important. And for me, at least as a community ecologist, not something that I had thought of as much kind of coming into this project. The duration of drought and age of the plant had opposing impacts on the diversity, especially for fungal groups. Drought can actually act as an initial introduction of stochasticity for AMF that kind of puts them on these divergent trajectories and increases their beta diversity across drought length, whereas longer droughts acted as a really homogenizing force for general fungi and bacteria. And then overall, we saw that bacteria and AMF are more responsive to drought, and this is more to drought intensity than general fungi. And so if you increase drought intensity, bacteria and general and AMF are going to be more responsive. Okay, so that's the water story. Next, I'm going to bring you two stories kind of together because they're in the same ecosystem. And so the earth story and the air story. And so this is going to bring us to New Mexico, which is where I did my PhD work. And we're going to think about plant soil feedbacks and ecotones under rising carbon dioxide. And so at the Sevieta LTER, which is where I work, it's in central New Mexico. It happens to be at this like really cool ecotonal region between the Chihuahuan grasslands to the north. So like the white yellow stuff, that's grass. That's really, really dry grass. And these Chihuahuan desert shrublands to the south. So this is a photo looking north. And these ecosystems meet at this ecotonal region, and change between these two ecosystems are really important to, for economic and ecosystem services reasons. So shrublands, a lot of people think about shrub encroachment. From kind of an ag point of view, locally, it's undesirable because that takes away grazing land, which is most of what these lands tend to be used for. From a carbon fixation point of view, like these different ecosystems store different amounts of soil carbon. So there are a lot of reasons to care about why shrublands transition into grasslands and vice versa. Kind of the mental framework we have for this at the Sevieta can be kind of broken down into this sort of what I think of as local interactions between grasses and shrubs and whatever else it is that they do. In my case, microbes, because I like microbes. So you've got these creosote bush, which are the dominant and really only shrub species, and black grama grass, which is the dominant grass species, right? They interact with each other via intraspecific competition and interspecific competition. And then where they exist together, they may also interact with each other via their soil microbial communities, via a phenomenon called plant soil feedbacks, where, for example, the microbes associated with creosote bush may have different impacts on its own growth and its own seedlings growth compared to the effect that black grama has on its own microbes and its own seedlings growth and vice versa. And all of this is happening kind of in a framework of elevated CO2, which is one of the factors that people have thought about for shrub encroachment. I'll get to that a little bit later. But one thing that I was really interested in for this particular ecosystem is it also affords us a really interesting way to look at the spatial extent of plant soil microbial interactions, because often you're in these systems where all these plants are growing all together and you can't separate them. And what I really want to think about is like, we know plants are sessile. Their kind of influence below ground only goes so far. We don't know how far it goes, right? And so these sorts of feedbacks, like how far do we have to get away from the maternal plant to actually have this happen? And does the strength of these interactions actually attenuate with space? We don't actually know, and it's really hard to quantify. And so I took this as an opportunity to actually try to figure that out as well. So the spatial extent of plant soil feedback is going to be the first vignette that I will talk to you about today. And so basically kind of the theme is consequences for plant soil feedback mechanisms and outcomes. So to measure the scale of plant soil feedbacks between these two species at their range limits, we devised this somewhat complicated experiment where we identified naturally occurring sort of resident plants, we're going to call them, instead of maternal plants, because we're not dealing with their seeds. So in this case, this is one of our creosote bush resident plants. And we set out these little PVC rings in which we germinated and grew seedlings of either the shrub species or the grass species at different distances away from it. And then these under the PVC rings, which you can't see, there are microbial treatments to try to isolate the microbial effect. And so we did this with a transplant control where basically we just dug the soil up, put it back in, nothing. The microbial feedback control where we dug the soil up, sterilized most of it, put it back in a bag, and re-inoculated with the soil that came from that site, just at 10%. And then the sterilized microbial exclusion where we sterilized the whole thing. And so this is kind of what a bag looks like when you dig it up. And here's the little tiny seedling you can see inside it. And here's us processing the bags at the end of the experiment. And so our response variables were maximum germination during the growing seasons. This ran for 2 growing seasons, and then above ground biomass of these little seedlings at the end of the second growing season. So the results that I'm going to show you, just to kind of be transparent, kind of follow this model. And so the model we have here is the identity of the resident plant, so either the shrub or the grass, the distance away, so these distances, and also the microbial treatment that is involved. We analyze the shrub seedling responses and the grass seedling responses separately. And then for germination, since there are 2 years, we're going to add a year effect. Okay, so that's kind of where those models come from. And then the results I'm going to show you only kind of highlight the significant explanatory variables, since 3-way interactions can be really difficult to visualize. All right, so what did we find? I'm going to start with the black grandma responses first, because those were the most interesting responses. And so these are black grandma germination data. And so this is the only one where we should have found actual spatial effects. And so first, what we're going to see here along the x-axis is the distance away from the resident plant. And then the panels are the 3 microbial treatments, the control where it's got everything, feedback where it's just got microbial feedback, and exclusion where it's got nothing. The resident plant can be either it's growing next to a black grandma plant or a creosote plant in the middle. Okay. And what we found first was a positive plant flow feedback. And so more black grandma germination near the conspecific resident. And you can see that by the fact that the yellow lines are higher than the green lines. And so there's higher germination for black grandma where it's growing close to a black grandma resident plant. However, that effect attenuates with distance from the resident plant. And so germination is the highest for these yellow lines where it's closest to the resident plant. And that kind of it slopes back down as you get farther away from the resident plant. However, we did not find significant effect of microbial treatments. And so you'll see that this negative slope occurs for all 3 panels. And so this effect might not be entirely driven by microbes. It might be something else. And there were no differences between the 2 years in germination. And so this was really interesting. Past work that I've done also have found positive plant soil feedbacks for black grandma, so that was good to kind of recapitulate. But this sort of like not specifically microbial, but kind of distance effect was like kind of puzzling to us. Next, I'm going to just show you real quick the biomass responses at the end of the experiment, where we still found positive plant soil feedback, so more black grandma growth near conspecific residents. However, the distance effect had completely gone away. There was no distance effect anymore, and still no effect of the microbial treatment. So at least that was consistent. Going to the creosote responses, and so these are little creosote seedling responses now, what we're seeing is that the strongest effect we had was the year effect. For whatever reason, in terms of like just the climate of the 2 years, we had much better germination the second year than the first year. However, we did consistently have negative plant soil feedback. And so there was more creosote germination near the black grandma resident plant. So the yellow points are higher than the green points. And no distance effect again. However, we saw that there was higher germination when root competition was excluded. And so in these 2 treatments where there was a bag, and so there was no direct competition from the resident plant root, there was higher germination overall compared to the control where we just kind of put the soil back in and put a collar on. So again, like so in this case, we were unable to capture any of the distance dependent germination effects. And one of the things that we're thinking about is there is potential that we just were looking at too close a distance for a creosote bush and for creosote responses. It could be the case that you might have to, drag out the distance away from that resident plant quite a lot more to see any attenuation of any effect, but we don't know. We were kind of going off of our best guess, and also what the site allowed us to do in terms of how much gap there were, distances between those creosote bush plants and nature. And then on the biomass responses, again, that negative plant soil feedback still held through to biomass. Again, no distance effect. And in this case, there was just highest growth when microbes were excluded, and this effect was specific to resident. And so there is some sort of plant soil feedback situation going on that may be facilitated by microbes, where we see a stronger negative plant soil feedback, so more difference between the yellow and green points when microbes are included, so in the control and feedback treatments. And so just to kind of give you everything all again, so that's just everything I just said, all in a table form, just to remind you. Some of the things that are really consistent are the direction of the plant soil feedback we've been finding. Creosote has negative plant soil feedback relative to black grama has positive plant soil feedback. But the other things kind of tend to go back and forth. We're not seeing like a super strong pattern. And so what does that actually all mean? Well, one thing we can do is actually sequence the plants and see if we can find a smoking gun or at least a pattern in the root microbes to show us something as to like, are we actually capturing like the right distance? Are things actually changing by distance? And the answer is yes, maybe, kind of, a little bit. Not very strong though, I'm just gonna say, right? And so these are the two plants. We got the black grama seedling, the creosote seedling, and this is the just like richness of root fungi. And we see that the patterns are all over the place. In this case, like I've left the significant slopes solid and the non-significant slopes are dashed. And so we're seeing positive slopes in terms of distance away from resident plant when we're in control. And then in feedback, we've got some negative slopes going on. As with exclusion, you might be asking, hey, didn't you like sterilize? This wasn't the sterilized, why are you sequencing it? Why does it have microbes? You know, this is a bag that's been left out in the field for 2 whole years. It might have started out sterile. It will not remain sterile, even though the mesh that we use is of such fine, it's at 0.22 microns. And so you actually cannot have fungal hyphae penetrate like through the mesh, but stuff is going to come in from the top. There's no way we can exclude top dispersal. But we're seeing very strange patterns. So this is where I'm just like going to wave my hands and tell you, I just started analyzing these data. So I don't have a lot of interpretation because it's kind of, I will say though, creosote and the shrub and the grass have very different microbiomes. So that's cool. At least we know they're different, right? So seedling species identity was the best explainer of root fungal composition. And you can see here by the clear distinction between the green and the yellow points. However, the one way I've tried to figure out distance structuring of the seedling root fungal community has not really, helped me. And so if you have ideas for analyses, I would love to hear them. So the way that I've thought about it so far is I can take these community matrices, I can calculate Bray-Curtis dissimilarity between samples, right? And I can compare the samples that are far away to the 0 distance. And if there is distance structuring, then we should see that the farther away ones are more different than the ones that are closer to the origin, which should result in a positive slope, right? More dissimilarity farther away. We see that for this slide. So these are the creosote resident seedlings in the control, but we don't see it in any of the other treatments. So I think what all of this together is showing us is that a combination of microbial and abiotic mechanisms could be driving these feedbacks, which have been quite consistent in terms of our ability to measure them from the plant's perspective. But we're still kind of in the dark when it comes to the mechanism. So it's maybe not necessarily all microbes, and it could be some other things. We don't have the smoking gun yet. We are working on the soil chemistry data to see if it's nutrients. And we are also doing additional experiments to look at other mechanisms that may drive microbial assembly below ground in this ecosystem. And so hopefully we'll have kind of more information as those experiments come to fruition. Okay, the air part. I want to talk a little bit about the impacts of elevated carbon dioxide on some of the mechanisms of plant soil feedbacks. And so I was talking about how all of this is happening within the context of elevated carbon dioxide, but what do we even care? I mean, that's everywhere. Why do we specifically care about it for this scenario? The reason why we care is for most shrub encroachment scenarios, a lot of it happens in dry, hot ecosystems. And in a lot of those cases, we end up with a scenario where you got a C3 shrub invading into a C4 grass sort of situation. And we know that these different photosynthetic pathways have different efficiencies when it comes to taking advantage of additional carbon dioxide as, you know, kind of the starting point for photosynthesis. And in our specific case, you know, the other usual suspects of shrub invasion isn't really an issue in this particular ecosystem. And so we don't have overgrazing at the site. The only things we have are pronghorn and invasive African mammals that people brought for big game hunting way back in the 1800s, but they don't like thrive. So we got a couple of them, but not a whole lot. And like, there's no real like fire issue. So these burn at kind of their natural fire return intervals, which is not very often. It's so dry out there that you don't want it to be crisping up and burning all the time, because these plants do not recover from fire very well. And so elevated carbon dioxide, why not? Let's think about it, right? And so we hypothesized that elevated carbon dioxide was likely to impact plant photosynthesis, carbon assimilation, and therefore their investment into root exudate carbon, and then because of that, microbial assembly. And so these were kind of our questions that we had. So how does elevated carbon dioxide affect the composition and diversity of root exudates, their rhizosphere, microbial diversity and composition, as well as biomass and plant soil feedbacks? And so my student Melanie took this on and designed a growth chamber experiment where we were able to actually manipulate carbon dioxide. So we had an ambient treatment and an elevated treatment. So that was 400 ppm. This was 600 ppm. And we grew the two plants either in live or sterilized soil inocula in soil collected from under plants that were either creosote plants or black grama plants, and either sterilized or live to try to kind of isolate that microbial component yet again. So all of these different treatments are fully crossed. And the response variables we looked at were plant biomass, the root exudate metabolome, rhizosphere fungi, and bacteria in this case. And so I'm gonna focus today just on the live component because it turns out that the sterilized component is a little bit wonky in that it didn't get fully sterilized one and then got recolonized kind of in a really weird way. And so we're just gonna focus on the live pots today. And so first, carbon dioxide has a really strong effect on plant root exudate metabolite composition. And so the way we kind of got root exudates is at the end of the experiment, we depotted the plants really gently, washed them off super duper gently, and then let them recover in this like root slurry for a little bit. And then kind of washed all of that off and then put them in pure nanopure water and just like agitated them on ice for 4 hours. And then all of that water got sent to, in this case, just a GCMS to look at the primary metabolites. And so in this case, we can see the CO2 treatment, which is shown in the different shapes here, has a strong impact, but the plant species of origin has the strongest impact on root exudate metabolites. Just to break it down a little bit, elevated carbon dioxide specifically decreased root exudate metabolite richness. So in this case, we have richness on the y-axis. You can see that for both plant species, the elevated carbon dioxide group has much lower richness in their metabolites. and also shifted in terms of the compound classes that we found. And so you can see already that the two plant species have very different relative distributions of the compound classes that we have here. But for example, when you go from ambient to elevated carbon dioxide, both species had declines in organic acid and sugar compound richness specifically. And these are like your delicious, delicious microbial food, right? Like microbes love that stuff. And so when we looked at the microbial composition, what did we find? We found that for bacteria, the results were pretty subtle. They were only very subtly different in terms of the CO2 effect. There was no effect of like where the soil originally came from, and some effect of the plant species that it was associated with. However, we saw a slightly stronger effect of CO2 on fungal composition, which had an interaction with soil source. And so this was a soil source by plant, by CO2 sort of like interaction here. And so what I'm like kind of seeing, or we're trying to decipher here, is that it does seem like we have a stronger effect of carbon dioxide on the black grama grass rather than creosote, which was the opposite of what we expected, given that creosote is the C3 plant. And so how does that manifest in terms of fungal diversity? So what we saw was that CO2 flips the relationship between soil source, so where that soil originally came from, and fungal diversity. And so if you looked at the ambient column here, you'll see that for both black grama and creosote fungi, that when they were inoculated with their home, so their conspecific soil inocula, they had higher diversity, and lower diversity when they were inoculated with the other species soil inoculum. However, that effect flipped when we went to elevated carbon dioxide, especially for black grama, which again had that slightly stronger impact on fungal composition as well. And so can we kind of link that to plant soil feedbacks in terms of plant biomass responses? Well, a little bit, because this is actually shoot biomass data, and the pattern looks weirdly the same. And so in this case, again, also carbon dioxide flipped the microbial and non-microbial effects of soil inoculum, potentially kind of priming us in a way for microbially assisted creosote invasion in an elevated CO2 future. So the reason I'm saying that is for both black grama and creosote, that in elevated carbon dioxide, they both actually do better in each other soils, in the away soils, right? And we know that in this particular ecosystem, that creosote has been increasing in density over the past like 20, 30 years. We have long-term data to show that. However, it hasn't actually started invading in terms of like moving its front further north. But at least the data we have shows that if it did start doing that, it should do quite well. And that also corresponds to some of the field data that we found. And so this is just to show the kind of fungal diversity data and the shoot biomass data side by side, which shows very similar patterns. I mean, I don't think there is a one-to-one correlation between fungal diversity and plant soil feedbacks. I think it's more complicated than that. But I think this is at least strong evidence that in this system, that fungi are driving probably more of the dynamics than soil bacteria are, where we don't see a whole lot of response in the soil bacteria. And so for this, kind of just to wrap it up, I think we were able to show that, kind of contrary to our own predictions, ECO2 affected the creosote bush and black grama pretty similarly. It decreased root exudate diversity and disrupted the relationship between plant hosts and their rhizospheric fungi, flipped plant soil feedbacks from positive to negative for both species, but we still need to establish any direct linkages between root exudate compounds, microbial communities, and plant soil feedbacks. And this is really difficult. And I was kind of speaking to a couple of people earlier. I feel like we're at a point in some of the work that we do in the lab where we need better statistics, honestly, that deal with multiple multivariate data sets and our ability to correlate them together. And so the last little vignette that I'll share with you all has to do with fire regimes and soil microbes in a longleaf pine savanna. So this is the fire story. So all of this work was done by my student, Michelle Henson, who graduated last year and conducted at the Jones Center in Itchaway. So this is in kind of southwestern Georgia. A few of you are nodding. You may be familiar with this site. Beautiful, beautiful longleaf pine savanna ecosystems here. And they're very serious about their fire there. Like they will burn and they will not stop burning. You cannot get them to not burn a unit. That's kind of where they're at. But they had this kind of interesting 20-year-long prescribed fire regime experiment where at these 3 different sites, kind of located not super close to each other on the property, they had been doing some long-term monitoring plots and then decided like to add a set of plots on the adjacent burn unit and then burn that instead of the maintenance regime, which is usually every 1 to 2, 1 to 3 years, depending on kind of how fast things are growing back, but in the dormant season. And so right now is actually our burn season. And so that would be the maintenance regime. And instead they put in a yearly growing season fire for the comparison sort of treatment group. Just the original intent sort of 20 years ago was to try to understand if that would better stimulate wiregrass growth and wiregrass reproduction. And so wiregrass is a species that is the foundational kind of understory species in longleaf pine savannas such as these. So all wiregrass here. And its reproduction is very fire responsive. And so they were trying to kind of generate more reproduction and they wanted to try to do just annual growing season fires. If that could really stimulate reproduction. And so they put in these long-term monitoring plots that have like 20 years of plant cover data in these plots. And so my student showed up, she's like, I want to look at fire effects on soil microbes. And I was like, great, let's go to the Jones Center. And they were like, well, we have this like really interesting fire regime experiment that we haven't done much with. So maybe you want to start there. And I was like, great. So really the first thing that we did that we just went in and just sampled all the plots. See what was going on, right? It's been going on for whatever it's been going on for 20 years. So there should be something. So that's exactly what we did. And we first looked at the plant data because that existed. And so they do see this really strong impact on plant composition. So the annual fires really shift that plant understory plant composition and actually decreases understory plant diversity. Okay, so there's lower plant diversity in the annual fire plots. And what we saw when we sequenced the soil microbial communities for fungi and bacteria is that soil fungi are much more sensitive to this change, long-term change in fire regime than bacteria are, right? And so here the annual regime is in red, the maintenance regime is in blue. And you can see strong shifts in plant and fungal composition, but like barely any shift at all in the bacterial composition, even though it is still significant. What was really also quite interesting to us is that it appears that annual fires homogenize soil fungal diversity, increasing the beta diversity. And so the kind of annual fire cluster here, all of these communities are much more similar to each other than within the maintenance fire. And that's really interesting because remember, all these points come from 3 different sites, right? And so this is aggregated across these 3 pretty far away from each other sites. But just the fact that there was annual fire really made them super similar to each other in soil fungal composition. In terms of what's happening in the phylum level, we mostly see a shift in Micromycota versus Ascomycota. We see way more ectomycorrhizal fungi, interestingly, in the annual plots compared to the maintenance plots. But in terms of like any like really light bulb moments when it comes to what's shifting in this little fungal community, we didn't really find any. It was more like a combination of lots of small shifts is what we're seeing. And then a second experiment that we decided to do was, well, let's pick one site and we really want to understand how a single kind of location responds to fire through time. And so what we were able to do is sample one site, all 10 plots through time, pre-fire, and then all the way up to 12 months post-fire. We were able to do this at 2 different depths and really get this like high resolution picture of what happens below ground as a fire comes through a site, which was super duper cool to me. And so what we see here again is these 3 communities respond in completely different ways, right? And so if we start with the fungal taxa, and we have 2 kind of different lines here because Michelle sampled both the surface soils as well as the subsurface soils for the microbial communities, with the hypothesis that the surface soil should be much more impacted than the subsurface soil, right, when a fire comes through. And so we did see that, but in a way that was surprising to us. And so this is the pre-fire. Immediately post-fire, you see that diversity just drop, right? For fungal communities in both the annual and the maintenance sites, but it drops more in the annual sites. That's one. 2, we were hypothesizing that the subsurface soils would be really protected. Those communities shouldn't shift that much. No, what we actually saw was that there was actually quite a big drop in those subsurface communities, and they did not recover fully in some cases, even at the end of the 12 months. So we're kind of thinking in kind of our future work about issues of dispersal limitation potentially for these subsurface soils. Whereas if you look at the bacterial species richness, you got fire and they kind of just do whatever they want. They're not really responding to fire in any way that we can actually figure out. You know, these patterns are mostly seasonal and it's just kind of changing through time, but not specifically in response to the fire event. Whereas for plants, because the fire happens in the dormant season, right? Especially for the maintenance one, you don't actually see much response until like it's starting to get into growing season. You get that like, you know, obvious increase in diversity and then drop off in diversity. Whereas it's interesting to me that in the annual fire plots, what we see is like a more kind of steady diversity throughout the year instead of this like stronger response to fire throughout the year. And so my guess is that is due to that compositional shift that has already happened, right? You have a completely different assemblage. They have different phenologies, different responses to fire. It's a kind of a more of a steady phenology throughout the year versus this like kind of more dramatic response to fire. And so just to wrap this little vignette up, we found that soil fungi are much more sensitive than bacteria to changes in fire regimes, and that there may be such a thing as too frequent fire if species richness is the metric that you really, really care about. But obviously, we also care about other things beyond species richness, right? So I'm not saying that don't burn, but that, you know, depending on what you're measuring and what groups you're kind of considering, there may be different optimal fire return intervals. And with that, thank you for listening. I want to thank my lab members and all of my funding sources and our wonderful collaborators, and solicit any questions if you have any. Thank you. Yes, go ahead.
The experiments between the creosote and the black rama were really interesting. So is creosote just allelopathic? Like, why does it not let specifics grow near it?
Yeah, that's a great question. I think we do see some evidence in our results that some of it is microbial. So there are probably host-specific pathogen buildup, but it is maybe allelopathic. There have been a couple of earlier experiments done in the 90s demonstrating some allelopathy, but mostly from litter, from creosote litter. So its leaves are well known to have a ton of secondary metabolites. It's very pungent. You can just smell it, like there's stuff going on. It's not easily herbivorized. But we don't know really much about root allelopathy in this species. There's not a whole lot of work done there. I think one of the few papers that looked at it basically just threw activated carbon into the soil to try to like soak up whatever might be there and then showed that there was kind of an effect. So I think there are indications of allelopathy. And so actually one of my students right now is starting a greenhouse experiment where we're going to investigate this using kind of litter tea and also some of these extra root exudates that we still have hanging around in the freezer. Yeah. Mhm.
Thank you.
Yes.
Great talk, Annie. You work in
Thank you.
one of my favorite places in the world. I think I have an idea for your Larry. Yeah.
Oh, yeah.
So Boodle is C4.
Uh-huh.
Creosote is C3.
Yeah.
You could collect respired CO2
Uh-huh.
away from each drop and measure the isotopic ratio of carbon and CO2. And then you could use a two-ended mixing model. You could basically have a metric of how much soil influencers from each type of plant ubiquitously across.
Is it sensitive enough to?
Oh yeah.
Oh yeah.
Yeah.
So like just soil respiration. You're talking soil respiration, right?
Yep. So soil respiration is going to come from roots.
Yeah,
Microbe community, but the base is going to be either the X phase or fine roots.
Right.
Or whatever. So
Right.
the base is going to be the same. And it's either
Right.
going to be -14 in the case of
Ooh.
C4 or -27 in the case of C3.
Well, that would be
It's like a perfect benchmark.
ideal. Cool. All right. Yeah, for sure. I want to talk more about this.
One grumpy,
No one suggested this before yet.
right, one
Yes,
grumpy old man thing.
Please
Look,
go ahead.
Nothing exists before 1990, right? In 1970, Larry was reported as the case example for allelopath. And then
Mm-hmm.
there was a whole literature into the late 70s, early 80s that it was water competition, that they
Mhm.
locally dry down the
Oh.
environment. And then there was a guy, another thing old, I can't remember his name, who was at UC Santa Barbara, I think, and they did root recognition experiments with malaria
Uh-huh.
and other species.
Yeah.
But it's like this whole set of papers, it's only in JSTOR's answer.
Okay.
Really-
I will double down on this search.
Yeah.
Thank you. I appreciate it.
But the required CO2 is way better.
Okay. I will take you up on that. Very cool. Thank you so much. Yes.
I'm curious, it seemed like a lot of the test sites for the fire regime study were near the edge on the boundary. What was it, what did it look like outside of the boundary? Because I feel like that would have some influence on the microbe community and all that. And then on top of that, how much does the water table influence the reintroduction of these microbes?
Yeah, you know what? I hadn't really noticed the kind of boundary issues here. I mean, obviously this one, I can tell you for sure that it's just all the same all around it. Here on the boundaries, I'm trying to remember, because like every boundary is slightly different out there. Like sometimes it just adjacent is just ag, that's it. But in other boundaries, it's just more woodland. I can't tell you off the top of my head, but that is a great question. We did take care to kind of where the plots are located are pretty center or like away from the edge. So there's at least that. Yeah. Mm-hmm. Sorry, what was the second part of your question? It like slipped my mind now.
No, you're fine. You know, thinking about in the vein of some of these being near the edge, how that influences water flow. and with
water.
You know, like the rate water table.
Yeah.
Mix and
Uh-huh.
recalculate places.
Yeah, that's a great question. I know water table is like such a big thing here. In talking with the folks who work out there, like they don't seem to reference it as much. I honestly couldn't tell you what my intuition is with that. What I can tell you is that we are really curious about the dispersal aspect. I have my newest graduate student is currently trying to design an experiment where we try to measure microbial dispersal via a couple of different vectors from both like aerial, above ground, and also below ground, whatever might be happening. So maybe I'll have something vague to tell you in a couple of years. Yeah, we'll see. I think it is really cool though. I really want to know more about microbial dispersal. And I think, you know, the days of everything is everywhere is long past us when it comes to microbial ecology. So we need to know. Yeah.
So I have a question online from Eric Minkus.
Yes.
Thanks for doing such a wonderful job of explaining several large-scale complex experiments. Can you share your feelings about the trade-offs between field experiments, garden experiments, and those done in greenhouses or labs?
Yeah, that's a really big question, and I think something that I kind of weigh the trade-offs of quite a lot. I love field experiments. Like if I can get away with doing something as a field experiment, I will. Because I do think that there are all these things that are happening in the field that are probably the most important if we, at the end of the day, want to apply whatever answers we have to the field, which, for ecology, that's mostly what we want to do. So for me, like if it's doable in the field, I would rather do it in the field. But I also love greenhouse experiments. And I think with a lot of these microbial type things that we're trying to do, it can be really difficult to get anything that's clean in the field, right? You saw that field experiment, like we can get treatment effects, but then we're not sure where those treatments are coming from because, you got these things that were originally sterilized, which were then recolonized by God knows what. And so I think for microbial inoculation type experiments, we generally prefer to do it in like the greenhouse or in the growth chamber if we would need really controlled environments. Another thing that I now talk to students about that kind of is just another layer of consideration is just risk. I try not to have a student's dissertation be entirely field experiments that take multiple seasons to run just because the risk is high, like something might not work out. You want to have a dissertation at the end of the day. And a lot of the field experiments we run, we actually do parallel greenhouse experiments that investigate similar predictor variables just as a backup. And if the field experiment works out, generally we get very similar results in the greenhouse that may or may not get published alongside, but at least it's like a nice backup in case something terrible happens in the field, which happens all the time in New Mexico. You get one bad year and everything's dead. So
one more question. I have a question.
Yeah.
First, in your first one, I noticed for your AMF
Yeah.
slide, it looked like your diversity, I can't remember if it was Shannon or Richness, but it looked like
Yeah.
super low. And AMF diversity is nothing like
Yeah.
just fungal diversity.
Yeah.
I was curious if you just given that it's also a greenhouse experiment, could you just look at each individual species or taxa of AMF, or are there still too many to do that?
I'll have to look back at that data set again. My guess is it's probably still a little bit too many for us to individually look at each taxon. And so just for context, I don't think I made this super clear. For that experiment, the soil that we used, it was just like 50-50 field soil and I think it was turfus for drainage. And the field soil came from like a sorghum field. It had like a crop sorghum field that we collected from. And so, that may or may not also be a factor in terms of low AMF diversity, because it did come from a crop field, but it also is relevant to AMF diversity for what it's worth. But so in the community assembly experiment, the data, which I didn't show, because we're still working on it, we were able to, from those soil, with INVAM's help, isolate 2 different species of AMF that we were able to grow out a lot more of, or they were actually able to grow out a lot more of, that we could then use in a subsequent experiment where we just did those 2 species and inoculated it at like different times relative to, like this classic priority effect type experiment on sorghum. And yeah, AMF has like really cool priority effect.
All right. Well, let's thank Annie.